Dear students, in this video we are going to discuss about three important fluids used in hematological experiments in physiology. One is Leishman stains, Hayem's fluid or RBC fluid, Dark's fluid or WBC fluid. So we can identify these are the bottles of Leishman stain. By definition, Leishman stain is a Romanowsky type of stain used for staining blood smears and bone marrow smears. So the root of Romanowsky staining can be traced back to the early 20th century when the Russian physician Dmitry Romanowsky sought to improve the visualization of blood cells under the microscope. So there are so many types of Romanowsky stains: Field stains, Dimsa stain, May, Garnwald stain, Leishman stain, Wright stains. So in this video we are going to discuss about the Leishman stain. All of these Romanowsky stains, they have a common characteristics. They commonly have typical two constituents. One is the eosin, another is the methylene blue. So eosin is a red dye. It selectively stains the acidic component such as cytoplasm, and it also stains the hemoglobin and eosinophilic granules. And after staining, it imparts a pinkish hue. In contrast, methylene blue, it is a basic dye. It targets the structures with a higher affinity for basic strains such as a nucleus, because nucleus contains the DNA, that is deoxyribonucleic acid, and RNA, ribonucleic acid. So acid will obviously attract the basic dye, and after staining it will provide a blue or purple coloration. The synergy between these contrasting dyes forms the basis of Romanowsky staining, allowing for the detailed visualization of cellular morphology. Now here we can see that there are two types of component present in the Romanowsky stain. One is the azure B, that is a blue cationic dye. So which is just like the a basic dye which will be derived from the methylene blue, and that stains the acidic structures. Acidic structures can also be known as the basophilic structure because it is attracting the basic dye. Whereas eosin Y, it is a red anionic dye. It is an acidic dye that stains the basic structures. Basic structures can also be called as acidophilic structures. Okay, so acidophilic sites are what are those? I have already told you: eosinophilic granules and erythrocytes, and after staining they will turn red or pink. Whereas basophilic sites will attract the basic dye, that is azure B, and it will stain the nuclei, that is lymphocyte, cytoplasm, and they will turn blue. Okay, now another important thing is there, that is the Romanowsky effect, that is a purple coloration. It occurs when the azure B, that is the blue cationic dye, that is from the methylene blue, and the eosin Y, they interact chemically to form a stable complex. You can see that one is taking the red stain, another is taking the blue stain, and the other one is taking the purple stain. So this purple stain happens when the azure B or the basic dye and the eosin Y or the acidic dye, they interact chemically to form a stable complex. Okay, this produces purple colorization in the chromatin, neutrophilic granules, and the platelets. Now regarding the composition, we have already told you that methylene blue is the basic dye. It stains the acidic components, or they are acidic components can also be known as the basophilic. So nucleus, RNA, and eosin is the acidic dye. It stains the basic components like cytoplasm and hemoglobin, and these acidic components are the basic components are also known as acidophilic. The solvent is the acetone-free methyl alcohol. Now methyl alcohol actually it helps in the fixation. It acts as a fixative. And why this methyl alcohol is acetone free? Because acetone may cause the lysis of the cell. Now results after application. microscopic appearance, you can see the RBCs will look as pink because they will take the eosin. WBC nuclei will become purple because here there will be interaction between azure B and the eosin Y. Platelets will be violet, and the parasites, the blue cytoplasm, red nucleus. What are those parasites that can be stained by the Leishman stain? It may be the Plasmodium leishmania, like this. And what is the applications? So it can be used for the differential WBC count and also detection of blood parasites. So this is the method by which we actually do stain. So first we make a smear, and smear must be completely dry before it is stained, because wet smear when stained, it does not stick to glass slide and gets wiped out during washing. So after drying, 3 or 4 glass sides of smear we should keep in our as a backup. So now after that, Leishman stain will be added drop by drop till it covers the whole smear. Now number of drop added is counted. Now stain should cover only the smear, and slide placed on the staining rack must be horizontal. This smear is fixed to the slide for the first 1.5 to 2 minutes, and this time is known as the fixation time. Okay, at warm room temperature, which may lead to evaporation, it results in thick syrupy layer of stain. That's why this may cause precipitation of stain. So during warm temperature, it can be prevented by addition of extra stain. Now fixation occurs because proteins are precipitated, preventing the film being washed off. At the same time, cells are preserved at the same chemical and metabolic state as they had at the time of staining. So this fixation is necessary, and it happens due to precipitation. After waiting for 1.5 and 2 minutes and allowing fixation to occur, equal number of drops of distilled water, same that of Leishman stain, are added to the smear. Water is added only and only after fixation is done, because it interferes with the fixation. Now what is the function of water? Addition of water after fixation causes ionization of the stain particle. What it does? It causes ionization of stain particles. This is essential for entry of stain into the cells. This is very important. Now ideally, instead of distilled water, we should use the buffer water. The pH of this buffer water is 6.8. It is the ideal pH for ionization of stain. Okay, after adding the distilled water, you can see also here a shiny greenish scum floating on the stain. It indicates proper staining. Now proper mixing of the stain with water is aided by the blowing air through the Pasteur pipette or dropper, and the mixture of stain and distilled water is kept for 10 minutes. Okay, initially the fixation time was 1.5 to 2 minutes. Then after adding distilled water, you have to keep it for 10 minutes. This time is called the staining time. At the end of the staining, the film should be gently washed in tap water. We should not directly strike the smear. Then it is dried and we can see the stain smear. So what is the end result? Methylene blue is a vital constituent of the stain. Stain is a synthetic dye belonging to the thiazine family. It is a basic dye, means it carries a positive charge. So the positively charged molecules has a high affinity for negatively charged cellular components. So methylene blue contains positive charge, so it has high affinity for negatively charged cellular components. What are those? The nucleic acid, RNA, DNA, acidic cell structures. Okay, so there will be the structures will look blue. So and these structures which are have higher affinity for this methylene blue, they are also known as the basophilic structures, acidic or basophilic. Now eosin is the acidic dye. It's characterized by negative charge. So obviously it interacts primarily with the positive charge in the cellular part. What are those? The cytoplasm and cellular proteins. Now cytoplasm, they will, after staining, okay, so cytoplasm, they have the affinity for the eosin. So these structures are known as the acidophilic structures. Okay, so they will stain the cytoplasm of the RBCs and also the eosinophilic granules. Now the cytoplasmic staining, eosin's affinity for the positively charged proteins in the cytoplasm results in pink or red coloration of the cytoplasmic regions of the cell. Now, so after staining, we can see these cells, we can identify the cells, we can see the neutrophils, their nucleus, multi-lobed nucleus, they are taken the methylene blue, where the cytoplasm is purple, and we can see the small lymphocytes or other large lymphocyte. The nucleus is totally almost it is covering the whole cell, and very thin amount of cytoplasm is visible at the central surrounding the nucleus. We also can identify the monocyte. We can see the eosinophil. In case of eosinophil, you can see that the eosinophilic granules, okay, so it attracts the eosinophil. That's why it is known as acidophil or eosinophil. So it attracts the eosin and it takes the reddish hue. Whereas the basophil, you can see basophil, actually basophilic, so it will take the purple in color. What are the uses? Leishman stain is used for peripheral blood smear examination to study the morphology of RBCs, WBCs and platelets. It helps to detect anemia, leukemia and infections. Differential leukocyte count, we can do differential counts of various types of WBCs. Detection of blood parasites, for example malarial parasites, that is Plasmodium species, and it also detects the Leishmania donovani bodies or LD bodies, okay, in bone marrow or splenic aspirates. Also used for trypanosomes in the peripheral blood. Bone marrow Leishman stain can be used for bone marrow examination to assess the cell morphology, maturation stages and abnormalities in hematopoietic cells. Platelet studies, so first is the blood smear examination, differential count, then detection of parasite, bone marrow examination, platelet studies, and veterinary and entomological studies for examination of protozoa, blood protozoa and insect vector infections. It can also be used. So these are the pictures where it is used. In the first picture above, you can see the malaria parasites are stained by Leishman stain. Below you can see the Leishmania donovani bodies or LD bodies, they are stained by the Leishman stain. And on the right side, that we usually do, we identify different types of cells. A is a RBC, B is a neutrophil, C filled, it shows the lymphocyte, D shows the eosinophil, E shows the monocytes, F actually shows the, and the last one shows the panel G, it shows the platelet, okay. Panel F is the basophil. Now how it is prepared? For the preparation of Leishman stain, we need Leishman powder and acetone-free methanol. So first weigh it, the Leishman powder accurately, 0.15 gram of Leishman powder we require. Add to it 100 ml of acetone-free methanol in a clean and dry bottle. Mix thoroughly to dissolve the dye completely. Allow the solution to stand for 24 hours at room temperature to ensure complete ripening and oxidation, and store in a tightly stoppered amber colored bottle. Protect from light and evaporation. Now these are the other stains, Romanowsky stains, Giemsa stains, Wright stains and the field stains. For our purpose, we actually just know the name of these stains and I have given details of their activity. So now come to the next stain, that is the RBC fluid or the Hayem's fluid. Hayem's fluid is an isotonic diluting fluid used for counting red blood cells or RBCs in the hemocytometer. The purpose of it is prevent RBCs from clumping or hemolyzing during counting, and it is invented by George Hayem 1878. Regarding composition of the Hayem's fluid, it contains the sodium chloride NaCl 0.5 gram, which maintains the isotonicity, sodium sulfate Na2SO4 2.5 gram, which prevents the RBC aggregation, mercuric chloride 0.25 gram, it acts as an antiseptic and preservative, distilled water 100 ml for as a solvent. Now the pH is kept 7.2, it keeps the RBCs intact for counting. Now how we do it, we know that this is on the right hand side we can see the RBC pipette. It has got 3 marking 0.5, 1 and 101, and one point make it clear that they have no units. They are the relative volumes. So we draw blood up to 0.5 mark up to in the RBC pipette. Immediately then we draw the Hayem's fluid up to the 101 marks. Okay, so how the calculation or dilution factor occurs, we will do it in the next video, where we will discuss about the 3 pipettes, RBC, WBC and the hemoglobin. Now after that, mix thoroughly by rolling the pipette between the palms for 3 to 2 minutes to achieve uniform suspension. Now after that, discard few drops to remove the fluid in the stem. Touch the tip of the edge of the newest chambers under the cover slip. Allow the chamber to fill by capillary action. No air bubbles or overfilling. Let the chamber stand for 2 minutes for RBCs to settle. So this is known as the charging of the newest chamber. After charging, you have to focus for RBC counting. You have to focus why for 40x I have already discussed. That is, we have to focus 1 millimeter square area at the center part. So we have to focus in the high power objective. Count the RBCs in 5 small squares, the 4 corners and the central one of the central grid, and calculate the total RBC count per cubic millimeter. So you have to express the RBC count per cubic millimeter. Without giving the unit, your result will be totally null. Now, what are the uses and precautions? Diluting blood for RBC count is in a hemocytometer. We do the total count of RBC by that. It preserves the RBC morphology. It helps in the demonstration fluid for hematology training. What are the precautions? Avoid air bubbles or overfilling. Mix blood and fluid thoroughly. Clean the chamber and pipette immediately after use. That is very important. And maintain correct dilution 1:200 and use fresh Hayem's fluid. So this is the RBC pipette. We can see that the blood has been drawn up to 0.5 and the fluid up to 101. So this is a method of charging. On the right hand side you can see that these are the RBC chambers containing 25 boxes, and after charging we get the count like this. Now the third and the last fluid is the WBC fluid or the Dark's fluid. It is our WBC diluting fluid, and its constituents are, you can see here, glacial acetic acid 2 ml, gentian violet 1 ml and distilled water 97 ml. So total 100 ml of fluid. Dark's fluid is diluting and lysing fluid used for counting the WBCs in the hemocytometer. What is the purpose? Destroys the RBCs and it stains the WBC nuclei for easy visibility. And what will be the appearance? Pale violet fluid due to gentian violet dye. So it contains glacial acetic acid. What is the function of that? It hemolyzes RBCs and fixes WBC nuclei. Gentian violet 1% aqueous solution. It stains the nuclei of WBCs. So the RBCs are destroyed, then WBCs are fixed and their nucleus are stained. Okay, glacial acetic acid will cause the hemolysis of RBC. It will fix the WBC nuclei, and gentian violet will stain the WBC nuclei. And the pH is approximately 2 to 2.2. So this is a procedure. On the right hand side you can see the WBC pipette. 0.5, 1 and 11 is the marking, and they also have no unit. So dilution factor is around 20. So mix the blood sample with Dark's fluid. Up to 0.5 you take the blood, and up to 11 you take the Dark's fluid, and the same procedure you follow as you have done in RBC counting, and charge the counting chamber in the reverse chamber. Allow the cells to settle for 2 to 3 minutes. Count the WBCs under low power. The RBCs are counted, total count of RBC is done under high power objective, that is 40x, and the WBC counting, total count, it is done under the low power objective, because here we are focusing total 3 into 3 9 millimeter square area of the Newber's chamber. So what are the uses for the total count of WBC? Demonstrating the WBC morphology, used in manual hematology, and precautions: avoid air bubbles while charging the chamber, handle glacial acetic acid with care, ensure through mixing of blood and fluid, use clean and fresh reagents. So this is, we can see the Stark's fluid containing the WBC pipette, and the second picture shows how when we are charging. After charging, it will look like this, and then we will focus the 4 cornered chambers: upper right, upper left, lower right, lower left, and under microscope we can see these are the 4 chambers containing the WBCs, and we have to count accordingly. Thank you.